Welcome to this month's Insaka. Insaka Talks invite speakers from different disciplines, and the idea here is to give people a chance to be exposed to an area that they may not normally attend a talk on. But I have to say, one of the most fun things for me to do is to try and spot possible links between the talks. So I'll see how well I shall do today. Today's disciplines are microbiology and paleontology. Our two guests are from Nigeria and Oxford, and the first talk will be on innovative approaches to understanding and tackling one of the most common childhood illnesses in Africa, diarrhea. Its persistence, I would think, in Africa does suggest knowledge gaps, which is what we're going to hear about today. And the second talk will draw attention to behaviors that could leave fossilized traces of destruction caused by humans. I have to admit, I can't wait to know more about these behaviors, the damage, and its extent. I'm also struck by the fact that these behaviors that we shall hear about do not emanate from tool use. So very excited about today's Insaka. Now about the speakers, Dr. Iruka Okeke and Susana Carvalho. Iruka is professor of pharmaceutical microbiology at the University of Ibadan. She works on genomic surveillance of antimicrobial resistance and also studies laboratory practices in Africa, an integral part of disease control and treatment. Iruka is a fellow of the African Academy of Sciences and currently advises the Nigeria Center for Disease Control. She is also on the Surveillance and Epidemiology of Drug Resistance Infectious Consortium Advisory Board of the Wellcome Trust. Susana is Associate Professor of Paleoanthropology and founder of the field of primate archaeology. Her work in West Africa on chimpanzees reveals how adept other non-human primates are at using tools. In East Africa, she does archaeological research in the Rift Valley. She's also the director of the Paleo Primate Project at Ngorongosa National Park in Mozambique, where she works on baboons, non-human primates, to understand human origins and behavior. As usual, the speakers will each give a 15-minute talk in succession, and the Q&A will be after the talk. So please do remember to post your questions in the chat box. And today, in case there are people who prefer to post questions anonymously, we have the option of using Slido, and my colleague has popped in the details on how to access it, including the code. So we shall move on to the first talk today. So the first talk is by Iruka, and her talk is titled Overcoming Barriers to Targeted Solutions for Infantile Diarrhea. Iruka, over to you.
Thank you so much, Watu, for the introductions. And thanks, everybody, for taking the time to hear what we're doing in Nigeria. And also, I'm also eager to hear about Susana's work as well. So today I'm going to talk about our work on diarrheal pathogens. I'm based at the University of Ibadan, which is in southwestern Nigeria. And I'm going to talk about work that we're doing that hopefully would lead to targeted solutions focused on specific pathogens. And before I did that, I wanted to acknowledge the fact that we know how to control diarrhea already. Diarrheal disease was a huge problem in the 18th and 19th centuries in New York, and that is no longer the case. And then in addition, in the current century, we've had new technologies that have overarching impacts on diarrhea if implemented. Things like improved water and sanitation systems, oral rehydration therapy, zinc, exclusive breastfeeding protects infants by giving them maternal antibodies and also protects them from pathogens that may be present in utensils that could be used to feed them. And we even have a well, but not extensively deployed tool that is pathogen specific. There's a rotavirus vaccine. So it's not that there aren't tools out there. It's not that there aren't broader tools out there. And I want us to keep this in perspective, because the work that we do, hopefully in the long run, will improve the management of diarrhea, but there are already existing tools that are underutilized that could help as well. So on the one hand, there's a big success story with diarrhea. In this century, diarrheal disease deaths and childhood deaths in general have fallen tremendously. Okay, this figure here from a Lancet review paper shows us that deaths per 1,000 live births has fallen in infant mortality has fallen from 77 per 1,000 to 46 in the period 2000 to 2013. And 6 per 1,000, the 6 per 1,000 drop is entirely due to progress with diarrheal disease. So diarrhea is a success story, and it's important to have that at the back of our minds. However, it's a success story that has started and is not complete, and success brings with it other problems. First of all, the success has been most seen in the first year of life. But children continue to die of diarrhea at rates that are not shrinking fast enough right after that year. Also, the children who survive because they didn't die of diarrhea are more likely to get diarrhea during their lifetimes. And this disease has important implications for the very young. If you and I get diarrhea, it messes up a holiday, maybe it even messes up your wedding. But if a young child gets diarrhea, it's not a 1 day, 1 week, 1 month incident. It's something that could affect them for the rest of their lives. Some of the long-term impacts of diarrhea include the fact that children who have repeated bouts of diarrhea in early childhood or persistent diarrhea, they have growth shortfalls. They have fitness impairments. This is medical fitness, not evolutionary fitness. They can have cognitive impairments, perform less well in school. And this all adds to the fact that their earning power in adulthood could be a lot lower. So we really want to do something to protect children against those bouts of diarrhea, even if they are going to recover. There are technologies out there that can give us more and better vaccines. However, those technologies are going to we're going to have to prioritize among pathogens. And for us to be able to do that, we need solid epidemiological information to understand which pathogens will give us the most bang for our buck if we make vaccines against them. There's also the possibility of developing new antimicrobials for persistent diarrhea. Persistent diarrhea and bloody diarrhea, by the way, are the types of diarrhea for which antibiotics are indicated. And right now, the existing antibiotics are not that useful because of antimicrobial resistance. There's also the possibility that hasn't been deployed in the clinic for diarrhea yet, but it certainly has potential, of developing antivirulence therapies. These are like antibiotics, except that they're not killing the bacteria. What they're doing is just stopping their ability to cause disease. And we believe that those sorts of tools might be less susceptible to antibiotic resistance. For those sorts of tools, you need good diagnostics so that you deploy the right tool at the right time. And again, good diagnostics requires us to have some information about what's causing diarrhea when. Without this kind of information, when we try to deploy tools, what we end up doing is working blindly, and then we don't get the maximum benefit from targeted tools. So many years ago, Dick Goran's group wrote a paper about the rest of the hippopotamus, it was entitled, essentially all those pathogens that cause most of the diarrhea that we know nothing about. And at the time, they were remarking on the fact that most of the diarrheal disease epidemiological information comes from studies that are done in returning travelers in high-income countries. So these are people who came from Europe or North America and went to Africa or Asia or South America on holiday, went home, took diarrhea home, and then back home they got diagnostics that figured out what was wrong with them. And then based on inference, it was assumed that these pathogens are what are killing and sickening young children in African countries and the countries that they came from. Things have improved since then. There's a bit more surveillance than there was then, but there are many countries where nobody's looking at causes of diarrhea. In most countries, only a few pathogens are being looked at. And then most of these efforts are short research-based efforts like mine, I must admit, that only look at disease in a small place over a short period of time. So we do need a lot more surveillance. One of the things that could help us is using different methods for surveillance, methods that will allow us get more depth, more breadth, more understanding of the pathogens. And genomics is really helping us to do that. And so what I'm going to share with you today is some of the things that we're doing using genomics and microbiology and epidemiology to better understand the causes of diarrhea. Now, my favorite organisms are E. coli. Hopefully, you've been told that E. coli is mostly a commensal. So most E. coli are harmless. If you're sitting in front of your computer and feeling good, then that means that you probably have E. coli inside of you. However, there's several types of E. coli that cause disease. And so we're particularly interested in those that cause or could cause childhood disease in African countries. I'm not going to go through the different types because even the typing has problems, but I will introduce those that I talk about. One of the things that makes E. coli so interesting to study from a biological point of view is that E. coli strains shuffle DNA around a lot. They can acquire information from outside, from other E. coli, from other organisms in pieces of DNA that are outside of their chromosome. And so this makes them an interesting population to study both genetically and evolutionarily. So what I'm going to do is share with you 3, maybe 2, depending on how the time goes, 2 of the questions that we're looking at very broadly in our lab, and tell you something of what we're doing and the approaches that we're taking, and importantly, how we and others in the field are thinking about this. First of all, as I mentioned, there's a rotavirus vaccine, which is wonderful. And there's actually a little bit of rotavirus surveillance surrounding deployment of the vaccine. However, we do need to understand which other pathogens cause diarrhea. Rotavirus vaccine does have impacts, but where it's used, children are still getting diarrhea. There are many bacterial and parasitic causes of diarrhea, as well as other viral causes of diarrhea that need better study. And also, there are real pathogens, suspected pathogens. Sometimes we don't know the difference between them. And so it's really important for us to be able to understand this better. In Nigeria, I'm a bacteriologist, so I focus mostly on bacterial causes of diarrhea. And we're particularly interested in understanding what causes diarrhea in the settings that we work with in Nigeria. There have been many multi-country studies in Africa. They typically exclude Nigeria. Nigeria is considered to be a difficult terrain to do research in, except for those of us who actually live and work there. But we also represent 1/5 of Africa's population. So if you don't understand what's going on in Nigeria, then you don't actually have a full epidemiological picture. And then most studies are based in teaching hospitals focusing on moderate to severe diarrhea. However, we're particularly interested in the mild to moderate diarrheas that are treated at home or in primary healthcare centers, because these are the repeated diarrheas that patients have that rarely see health facilities and that can impact them lifelong. There are several types of E. coli that cause diarrhea that are insufficiently defined, including my favorite, enteraggregative E. coli. And so we want to be able to come up with genetic definitions of these pathogens so that we know what we're working with and what is likely to be a pathogen and what's just a bystander. So what you can see on your slide here in my secondary school colors, blue and white, is a eukaryotic cell with a big nucleus in the middle. And the rod-shaped bacteria adhering to it are enteraggregative E. coli. And enteraggregative E. coli gets this name because of this stacked brick pattern that they form around eukaryotic cells. So the stacked brick pattern is characteristic of enteraggregative E. coli. If you see this in the lab, you say, okay, I have an enteraggregative E. coli. But if you've looked at these for a while, what you see is that this is a phenotype that could be, and we now know is, convergent. That is to say that different properties, different genetic characteristics can give you this same phenotype. And that means that this category as presently defined is very heterogeneous. So a very brave student of mine, Bisi Akinlabi, decided that she wanted to look at enteraggregative E. coli outside of central Ibadan, which is the dark teal in the middle. And we're actually looking at it in Northeast, where I put a box, and we're working with primary healthcare centers. They're looking at children with diarrhea and looking at healthy children in a case control design. And to cut a long and continuing story, by the way, she hasn't finished, continuing story short, what Bisi finds is that she finds the same number or proportion of enteraggregative E. coli in children with diarrhea and in healthy controls. And this is actually a common feature of many epidemiological studies. And the feature arises again from the heterogeneity. which we can now break down because she's sequencing her strains. And we can look at different factors that cause either the adherence or the diarrheogenicity and ask, what do these different factors, how do these different factors separate by diarrhea and controls, so that we are getting closer to getting a genetic definition. The other thing I should mention is that most children by their second year of life actually, experience one or more episodes of enterogative E. coli diarrhea. And so we're really interested in understanding, is it just diarrhea that's important? And it turns out that one of the multi-country studies that has just finished, a MALED study, is showing us that diarrhea isn't the only thing. Some children may have an enterogative E. coli infection and not get diarrhea, but it impacts their growth potential. It impacts whether or not they become malnourished in the future. Then a second question, number 3 on the slide, because I skipped one. A second question that we are looking at is, how common are outbreaks? When I was a PhD student, I was doing a short epidemiological study amongst adults. And we found that in that study, in week 10 of that study, there was an outbreak. And we thought, oh, this is really strange. What a coincidence that this outbreak occurred while we were performing our study. However, we now know that outbreaks are pretty common. And the only reason why we don't see them is because we're not looking. If we had routine surveillance, then we'd be able to see these upswings in diarrhea. And indeed, Bissy has seen these upswings as she's looking at diarrhea in the primary healthcare centers. And there's one example here that I've marked up in September 2016, when she had a rush of cases. We don't see very many cases in a given day, but in a couple of weeks, she saw very, very many. And when we looked at them, we found that a different category of diarrhea causing E. coli, enterotoxigenic E. coli, was responsible for this outbreak. And we're now looking at lineages of enterotoxigenic E. coli that we find, and then looking at what is special about those lineages that are found from within outbreaks versus those that are found more generally. Okay, I'm going to end here because what I'd really like to do is leave plenty of time for discussion so that we can talk about the questions that you have. I can't thank everybody in my group, nor can I thank all our funders, but I do want to thank the MRC DFID, which funds our diarrheal pathogen research. I shared Bissy Akinlabi's data with you, and the other people working on the diarrheal study are David Elshama and Ife, looking at water quality. I also want to thank Jalade that helps with our logistics and therefore gives us time to spend in the lab. A list of collaborators on the right hand of the screen, and looking forward to Susanna's talk and then your questions. Thank you very much.
Thank you very much, Iruka. And we'll come back to Iruka with questions shortly, but please pop your questions right away if you have them. And as she indicated, and also both Susanna, they're very keen on having a vibrant discussion, and no questions are stupid questions. So please post your questions. Remember, there is this slide, post them anonymously. And in the meantime, we're going to move on to Susanna's presentation, whose title is 15 years of tracing invisible steps in the technological evolution of early hominins. Where should we be heading next is the question. Susanna, over to you.
Thank you. Good afternoon. I hope you can see my screen. Thank you, Iruka, for a fantastic talk. I will start by saying before the talk that there are no topics that are impossible to breach. And while you were speaking, I was actually thinking that we have colleagues here in my group that have studied Nigerian chimpanzee tool use in terms of insectivory, the use of perishable tools to access insects. But also interesting, I have students starting a project on self-medication in wild chimpanzees. And so with all the health related potential discoveries, even for applications in humans, right? And trying to understand with climate change and all the changes in habitat, how these things may be affecting not only the consumption of these resources, but also the rate of certain, or the prevalence of certain diseases. So certainly, actually quite a lot to bridge and discuss together potentially. Now, moving to the topic of my talk, and I want to thank really AfOx for the initiative, all the colleagues here for the opportunity. The title is quite ambitious, and you can imagine 15 years of work in 15 minutes is quite a challenge. So I'm going to present, obviously, an abbreviated version. Excuse me, an abbreviated version of what has been a field of research that has led to a number of discoveries. Most of them are still ongoing, and then picking in on what Watu was saying. In the end, I will suggest some directions forward, including potentially focusing our efforts on other behaviors that may not be just technological behaviors. So my interest is really in the origins of technology, which is believed to have marked a major adaptive shift in human evolution. Okay, so understanding the evolutionary processes that underlie the first human adaptations to tool use and the following processes that led to our species being the only living primate that is fully dependent on technology, or we think so, is really one of the most stimulating topics of research of present day archaeology and human evolution. New fields of research have been founded over these past 15 years, including primate archaeology, what we could call Pliocene archaeology, so which is an older archaeology, during this quest to find out how old technology actually is, where it originated, and who were the first tool users. Historically, the vast majority of the information we have on this topic actually comes from the study of stone tools, which most people know what I'm talking about, tools that manufacturing is generally believed to be a uniquely human characteristic. This was the belief well into the 1960s. The production of these stone tools was linked first to the origin of the earliest hominins known, being thought to have co-evolved with traits such as bipedalism. It's something that was proposed by Darwin, or hunting, scavenging, and later to the evolution of our genus Homo, and also with accompanying increases in brain size. As a result of some of these breakthroughs in the field of primatology and more interdisciplinary research ongoing, actually there is a paradigm shift in the belief surrounding this uniqueness of human technology. This is what I'm going to be talking about today, and I'm going to be talking about that the possibility or the discovery more recently of napped stone tools and cut mark bones that are dating to about 500,000 years before what we thought was the evidence of tool use for our species, are giving rise or gave rise to the possibility that also earlier, previously unconsidered human ancestors or even other extinct non-human primates could have been responsible for the inception or origins of tool use and manufacture. I'm going to abbreviate my introduction here because I am slightly afraid of not having time, and more focus on the key questions that we have been trying to answer in the past 15 years, more or less. How old is technology and how can we know how old it is? Who are the first tool users? What is the comparative evidence that we can use to shed light into these questions? What tools likely composed the first toolkits in the past? What selective pressures may drive forage on the go type of tool use, which is what we see in the present day, for example, in wild chimpanzees, in which led to the obligate tool use and to what we think is the hominin technological dependence of today. What about other primates? Are they actually also dependent to some extent on the use of technology? And finally, where should we be heading next? Just like Iruka, I will not have time to go over all the questions, but hopefully will give you a sense of where we are in the most important ones. So, how old is technology? Questioning theoretically if there will be anything predating what we call in archaeology the old one, really only emerged less than 20 years ago. The first data supporting this idea that tools could be older than the 2.5 million that we thought they were would not correspond to the origins of technology was actually published less than 10 years ago in some of the papers that I reference here. So it's a very recent event that we are starting to accept that the old one is older than what we thought, and that there is something out there that may precede that type of technology. Looking at this diagram, just to give you an idea, an historical perspective on the number of hominin species, so our, in this case, extinct direct ancestors that have been found and published over the past 60 years, you can see a clear trend in the figure. So the first 40 years of research show or revealed us 7 different species of human ancestors. And after 1994, when there is a surge of discoveries and work in Africa, you see that a variety of hominin species are discovered, and that trend increases sharply after 1994. What happens though, if you try to do the same exercise that we are doing here with hominin species, plotting the old one or pre-old one archaeological sites that are known? And this is what it would look like, of course. You discount here the discovery use prior to the discovery use of the Potassium argon methods to date sites. So the 1960 date there refers to that, to the discoveries that were made before we were able to date anything older than 1 million years. It was just not there before, and it was first used by Louis Leakey in Olduvai Gorge. So again, what you see here is that in the past 20 years, you see an increase in the range of dates, and that is due to researchers now looking in deposits, in geological deposits that had previously been excluded from archaeological surveys, because it was basically assumed that there was no technology prior to the old one at 2.5 million years. And there was no technology in principle associated with non-Homo species. So it was the idea that we were very unique in terms of a genus, and that part of that uniqueness was our tool use technological ability, okay? So, okay, when you look here, what we knew in 2006 about technological origins and before the old one, you can see on your left how old and how many sites and deposits we have published with the oldest assemblages. Here we are talking about stone tools. And then on the other side, you have the hominins that were known at the time of these discoveries, okay? And if I change this to show you what we know in 2021, then in terms of the time depth of dated archaeological assemblages, you can see that we have now plenty of sites documented for the old one between 1.8 million and 2.58 million, including sites of Gona and the newly published site in 2019 of Bokol Dora 1 in Ethiopia. After that time of 2.58, the record is still very minimal, but evidence is accumulating, indicating that the old one probably has deeper time depth, and that other technological complexes may exist at around 3.4 million years, which is before genus Homo emerged, okay? If we try to place a confidence interval following the idea that we also use when we date fossils of a first appearance date and last appearance date, then it would be really very reasonable to assume that all the archaeological records are out there waiting to be found. And those could go back to 4.5 million years or even older. So one of the big questions. On the right side of the diagram, like I said, gives you a good idea of the variety of hominin species present over this time frame, plus the new hominin species that have been discovered after 2006. And in theory, we could consider all of them candidates for tool making and tool use. However, it's important to say that so far, no direct association between findings of artifacts and hominin fossils has been achieved apart from our genus. So in better words, direct association between artifacts and hominin fossils remains restricted to genus Homo, but we postulate that this is just a matter of continuing to do work and being lucky to find more hominins associated with these assemblages. Okay, so moving on to who were the earliest tool users, which is has been one of the quick questions also around the origins of technology and our work. Let's look at this diagram again to see what we know now about primate tool use and non-human primate tool use in terms of what we have documented via archaeological excavations and also using standard dating methods, particularly the papers by Aslam et al. 2009, and then in more recent years presenting excavations, systematically done excavations with dating well established, giving a date, for example, for genus Sapajus of 3,000 years in terms of the assemblages that have been excavated and dated. Much more recent time for macaca, so for long-tailed macaques in Thailand. And for Pan, for chimpanzee archaeological sites, we have also well-dated archaeological excavations pointing to 4,300 years at least of time depth into the stone tool used by chimpanzees, okay? So, but what we can see here that is important, I think, is that tool use in the wild is now documented for all great apes, albeit with very different frequencies of tool use. So, for example, for gorilla, it's quite anecdotal, and in Pampaniscus, it relates mostly with tool use that is not for extractive foraging, but for other purposes. Moving on to was the Pan Homo last common ancestor a tool user? So if we think how old is tool use and who were the earliest tool users, if we consider the possibility that tool use is a shared trait by Pan and Homo, and then we could postulate that potentially the last common ancestor between Pan and Homo could have been a tool user already. What we have in this case, I'm not going to spend time discussing if Pan is a relevant model. In this slide, there is the argument there is a long standing discussion about how did the last common ancestor Pan Homo look like, and we don't actually know because we haven't found it, with a surge in the past decade of researchers arguing that Pan may not be the best model to know or to how the last common ancestor looked like or behaved. This had a lot to do with the discovery of Ardipithecus ramidus, which was a very surprising hominin published in the 90s that shows many derived anatomical features, along with the argument that there has been as much molecular evolution in the gorilla and chimpanzee lineages as in the hominin lineage. However, more recently, we are indeed experiencing a return to the idea that plenty of lines of evidence, including comparative molecular and anatomical data from extant apes and fossil evidence, can be used to reconstruct when the LCA of chimpanzees and humans lived and what it looked like. And this paper I referred here from Pilbeam and Lieberman, a recent one, makes the claim again that actually at the moment, the evidence is suggesting that the LCA was morphological somehow like chimpanzee and gorillas, and probably much closer to a gorilla chimp size than chimpanzee size than gorilla size, which I think gives us good grounds or reasonable grounds to say or to be back to Pan chimpanzees as a good model to understand the morphology and behavioral adaptations of the last common ancestor. And we have published some papers in the past year where we do make the argument that we think tool use can be as old as this last common ancestor. Whether you believe or not that this LCA that may have lived about 8 million years ago, around that time, had the capacity to use or make tools, obviously ultimately boils down to how much homoplasy one is ready to accept among sister taxa. But what we do know is that complex tool behavior and including variability in cultural traditions across groups and habitual tool manufacture is a universal among chimpanzees and humans. And this is not something you can say about any other living primate, and has now been documented even in a semi-wild context for bonobos. So as such, I think I make the argument that it's unlikely that such an event would have evolved in parallel between sister taxa to the extent that became now fixed in all populations of both lineages, right? So I think it's very possible that indeed the last common ancestor was a tool user. The bigger question would be, can we go that far back in time to find and recognize tools that are that old? And that's a completely different question that would be a different talk to answer, but I am happy to answer any questions that may emerge during the question time. What is the comparative evidence that we can use in this field of primate archaeology, where we work in a very, very interdisciplinary, with a very disciplinary approach? Going back to the question of the origins of technology and when tools first appear, we need to consider any adaptation and selection pressures that may have driven the use of technology, right, in human and non-human primates. And this is when ethology, so animal behavior, and recording extant primate behavior in tandem with looking at archaeological and fossil data becomes really a major asset, I think, to make visible behaviors that cannot fossilize and will never fossilize. This is an extensive list of papers, but this is just to say that in general, our comparative evidence that we use to make these inferences about the earliest tool users and how tool use may have evolved comes mostly from anatomical, cognitive, paleontological, and behavioral data. I can say that all the lines of evidence so far point to or do not raise the possibility that any of the extinct hominins or even a less common ancestor would be unable to use tools or would be limited in any of these aspects to use tools. What tools likely compose the earliest toolkits? There has been, again, quite a discussion about this in looking at modern data, not only from non-human primates, but also from modern non-industrialized human populations, and considering general availability, distribution of resources in the environment, plus the cost of obtaining the resource to make and use a tool, most likely we propose that perishable materials would have been the main target. So not stone tools, but perishable organic tools. The advent of what we call the archaeology of the perishable is actually undergoing as I speak by Alejandra Pascual Garrido, Catarina Almeida Warren, and others, and will likely be the most important development in this field of research in the next years. The type of tools likely composed that likely compose the earliest toolkits, as I said, come from this idea that they would be mostly perishable, come from different lines of research, like I said, so hunter-gatherers, but also the fact that non-human primate tool use is mostly, across all species, a non-perishable type of tool use. So lithic is very, very rare, and most tool use by non-humans is performed with perishable organic tools. I'm approaching the end. Please bear with me. Where are we heading now? So primate archaeology is continuing to use observation of animal behavior to create referential models of tool use. This is something that has developed quite a lot and is ongoing. The study of tool use across different taxa that we have been doing really is allowing us to say something new about the origins of technology, about convergent evolution in technology, because obviously that's what we expect is happening when we look at more distantly related species like the capuchins or the long-tailed macaques using tools. And more importantly, is allowing us to to understand the behaviors associated with tool use and place them in an ecological and social context, which is something that hasn't been done before. Other lines of research also are tackling the cognitive abilities necessary for tool use, and are looking or found social transmission of tool use and inter-community conformity of tool use in non-human primate species. These were very important discoveries also of the past 15 years. Novel analytical tools have been developed and are undergoing. These analytical tools are essential if we want to actually be able to find things that we haven't been trained initially to know what they look like. And in this case, we applied some methods that had been used for archaeological assemblages to analyze chimpanzee stone tools. This is a paper we published in 2015. Studies showing and telling us about selection in tool sets. So telling us that chimpanzees select stones based on size, weight, and certain types of material, and that they reuse systematically specific combinations of stones have shedded some important light. And not only stone tool use, but similar studies done with termite fishing tools by one of my students on tool sets and selection, again, showing that chimpanzees select specific plants and parts of plants to make their tools to perform the termite fishing behavior. And finally, my last slide is to say we have been doing a lot, and I'm sorry I went a little bit over time. We have been doing a lot over the past 15 years. What and where should we be heading next? I think the suggestion is that we need to continue explore further technological behavior within the primate order. And this is independent of taphonomic issues. So this is independent that we are looking at tools that we think we assume are not going to fossilize, but that is not necessarily the case. And we have work undergoing that we hope will show that soon. Then we need to record non-technological behaviors. There are a range of behaviors at the moment, including bark stripping by primates, turtle smashing to eat the turtle, crab eating, a number of behaviors that are not tool use behaviors, but they may be leaving important traces in the landscape that could fossilize, especially when we are talking about robust wooden material, and that nobody has looked at, but right now we have colleagues starting to look at this. We need to revisit archaeological assemblages that have been recovered and assigned to hominins or human ancestors, but they may actually lack the context and they may lack the hominin association to make those claims that everything we are finding specifically in the Rift Valley in Eastern Africa has to be hominin. I think it's time to give a step forward and say something that may sound crazy at the moment, but maybe it's not going to be in 10 years, that other primates may be leaving behind very important assemblages, and we may be confounding some hominin with non-hominin assemblages. We need to develop new analytical tools that actually can allow us to identify material that we haven't been trained to find originally. And finally, we need to survey older deposits in different locations and get out of the usual locations of the Rift where we work all the time to look at the oldest technologies and use new methodological approaches that can help us optimize finding locations that are potential locations for fossiliferous deposits and archaeological sites. And we also need to improve in terms of behavior what we know of longitudinal records of the ontogeny of primate tool behavior. And we now have amazing records of decades storage across labs in the world, most of them not being processed yet because we lack the power and the resources to process such huge volumes of data. And a lot of these things are being tackled by different students and research in my lab, but others are still waiting to be done. I want to thank everyone that has helped me, my collaborators in Africa, Oxford, and visit please our lab if you want to know more about our research. Thank you very much.
Thank you, Susana, for that very persuasive presentation about non-humans and how they can inform technological evolution. I'll go straight to the questions because I can see there's quite a few that have come in. And I'm going to start with one for Iruka. You said something about outbreaks and people getting surprised, and it turns out actually it's because that systematic surveillance doesn't happen. And what I wondered there is, in different parts of Africa, there's some level of continuous surveillance that happens. And I wonder, to what extent is it very selective, in the sense that maybe it's focused on what are seen as very threatening diseases like Ebola and so on? And is there something about the assumptions that people make in terms of where they focus their attention? Because clearly, if diarrhea doesn't exist in other parts of the world and continues to exist, there should be mechanisms that focus on surveillance issues and so on. So maybe you can tell us a little bit about that. And I would like to link it with two questions that actually come back to the question of surveillance. And one of them is talking about if it's part of your lab work to maintain support for continuous surveillance of diarrheal pathogens. And the other one is saying, what are the main challenges of routine surveillance of diarrheal pathogens? Iruka.
Hey, thank you, Watu. There's some really important questions there. Now, it's true that there is some continuous surveillance in Africa. There's not very much of it, and it tends to be focused in specific places. The good news is that it's growing. 10 years ago, most African countries did not have a public health institute. So public health institute like the US CDC or the UK Public Health England, that's the agency that's responsible for continuous surveillance. So many countries a decade ago didn't have one. So if anyone was doing surveillance, it was researchers, KEMRI, MRC Gambia, places that had long-standing research institutions often took that up. But very often what you found is that in many countries, there was no pathogen-specific surveillance. Now, there's a Ministry of Health. The Ministry of Health collects data from health facilities. But if those health facilities are not using laboratory diagnostics, what feeds up is information about the syndrome. That, oh, a lot of people have diarrhea. The people who had diarrhea this year are more or less than the people who had diarrhea last year. But that doesn't tell you how many people had Shigella dysentery versus enterotoxigenic E. coli diarrhea versus enteroaggregative E. coli diarrhea. And because diarrhea is caused by so many different pathogens, it means that when one pathogen goes up and another comes down, if you're just looking at the syndrome, you're actually not seeing very well. It's really a blurred vision. Now, in terms of ongoing surveillance, yes. We are actually now working with the Nigeria Center for Disease Control, not specifically on diarrheal disease. We're working to build Nigeria's antimicrobial resistance surveillance system. This does include some diarrheal pathogens, but not all of them. And we're not the only ones doing this. A number of us in academia have realized that, our academic efforts to study a particular disease in a particular time and place is not the way to get sustained surveillance data. And so what we do is we partner with the National Public Institute, in our case, Nigeria Center for Disease Control, and help to build it up so that if we start something, it can continue there. So we're doing that for antimicrobial resistance. It does include some pathogens like Salmonella and Vibrio cholera, but certainly not many of the pathogens that cause childhood diarrhea. And then just to say that everywhere in the world, many of these pathogens that cause childhood diarrhea are not really looked at because they're difficult to find. You need genetic or genomics tools to find them. They're expensive to detect. If they're not that common, then it's not cost effective to look for them routinely. In places like Africa where they are common, it's still cost effective, even though it may be expensive to look at.
I'm going to stick with you a little longer, Iruka, just to follow on some of the things you said. So one of your slides showed a potential relationship between diarrhea and longer-term metabolic diseases. Could you say a little bit more about that? And you just mentioned genetics just now. So given the genetic diversity, being enteroaggressive a feature of E. coli, or does it reflect the host environment?
Okay, so let me think off the top of my head. I may have to come back to the first question last while I'm thinking. The genetic diversity in enteroaggressive E. coli really stems from the fact that that category is not a real category. And I'm sure many people in the field, our field is really small. I mean, we can all sit around a dining table. Many people in the field will be shocked to hear me say this, but I think we've got to the point now that we're getting genomic sequence, that we understand that what it is a collection of different things. Some of those things are actually really dangerous pathogens. Those of you who are old enough will remember in the early 2000s, I think it was, there was a large outbreak in Germany that spread around Europe of a diarrheal disease. That was an enterrogative E. coli. So we're not talking about something that isn't pathogenic. There are lineages that are pathogenic. There are strains that are pathogenic within that big category. But there are also some lineages, some so-called virulence factors that we find more in healthy people than in people with disease. And so what we've got to do is to sort that mess out and then redefine the category genetically. That is to say, not that enterrogative E. coli causes disease, but E. coli that have this feature, and that feature cause disease, whereas E. coli that look very much like it, but don't have these important features, don't, essentially. I'm still blanking out on a specific metabolic condition that arises from repeated bouts of diarrhea. They are there in the literature. If I remember them before the end of the discussion, I'll come back to you. But when you talk about heterogeneity, yes, there are heterogeneous pathogens, which is what I study. The host response is also heterogeneous. So that means that if I take a pathogen that does cause diarrhea and I give it to everybody, not everybody will get sick. So it's a mix of both things. You've probably seen this in COVID. Not everybody who's exposed to COVID actually gets sick, even though some people are infected. And the same is true for diarrheal pathogens.
Thank you. Thank you for that. So, Susana, now, so there's one, basic question just to get things right. Can you tell us exactly what technology means in this context before we carry on with the broader discussion of what you just presented?
Yeah, so it means I'm talking about tool use and tool manufacture, and we're talking about artifacts, so objects that are used, detached from your body to achieve a goal that wouldn't be otherwise possible to achieve. Either it's extractive foraging because you want to get a nut from a kernel, and so you're using a stone to break up to crack the nut, or because you're using a leaf to clean yourself. That's one of the many types of tool use we see in non-human primates, so that you will be using a tool for hygiene purposes. So when I talk about technology, I'm talking about all these different types of different technologies that ultimately in the archaeological record, we call them technocomplexes. For example, the old one, you would say it's a technocomplex because it can be systematically described and recognized as being technology that is composed by 4 different types of tools that normally show up together, right? And so there are many different technocomplexes across the archaeological records. The old one followed by the Acholian, etc. And in non-human primates, we are yet to describe actually techno-complexes in the same way, but we are talking about tools. And the thing is that there are tools that are modified by use. For example, the case of stone tools in chimpanzees, those stones are not manufactured to be used. They are found and used, and then when you find them without the chimpanzees using them, you can tell this is an anvil and this is a hammer because it has been used for a long time and it shows modifications because of the percussion activity, but it was not a tool that was made for the purpose. And then there is the tool manufacture that you see, for example, in termite fishing in chimpanzees, where the object is modified prior to use. So it's made to be fit a shape to fit a purpose. So is this helpful?
Yeah, that's fantastic. And based on bonobos and chimpanzees and their different tool use, which one do you think would provide a better model for the last common pan-human ancestor, and why?
That's a very good question. And I would say both. Because if we are talking about, yes, if we want to model the less common ancestor, pan Homo, then we need to be looking both at pan-paniscus and pan-troglodytes. Obviously, it's a very long debate and discussion about why bonobos show overall much less tool use in the wild than chimpanzees do, and why do they not have much extractive foraging. So most tools that bonobos use is not actually to access food, compared with chimpanzees. Kat Coops, my colleague who is in Cambridge, has done quite a bit of work trying to look at the differences between the bonobo and chimpanzee tool use. And she has actually found very interesting data looking at infants and development and playing with objects. And she proposes that pan-troglodytes has potentially an intrinsic predisposition for tool use. So you see infants manipulating objects much earlier, much more frequently than you see in bonobos. So bonobos play a lot, are a very playful species, but not necessarily as much as chimpanzees when you look at playing with objects. I think if we want to understand, especially the ecological drivers of tool use, the bonobos are a very puzzling case, of course, to look at. Why is it that in chimpanzees you have tool use as a universal in every single community that has been recorded across Africa, and in all of them you have some form of extractive foraging, even if it's just the using of leaves to drink water, which is a universal across chimpanzee populations. But in bonobos, you have such a different case. And some people have said it's just potentially density of individuals in terms of the availability of resources, which means they don't have the need to use tools for extractive foraging. But that remains to be further tested. Of course, where the bonobos are living is quite a difficult place to carry out work systematically. There has been a lot of gaps in that research, but it's ongoing, and and we look forward to know more about the bonobo tool use.
And very quickly, I'm very fascinated by a question here about wooden tools.
Mm-hmm.
They would not normally fossilize.
Mm.
But are there exceptional examples, and if so, how old might they be?
Yes. So yes, the short answer is yes. I think it's true, they would not normally fossilize. So if you think about the twigs used by chimpanzees for termite fishing, the likelihood of such tool fossilizing is extremely low, almost zero. However, certain contexts of tool use, for example, if you're using tools in a cave, and that cave for some reason is sealed very quickly, we have some discoveries. Actually, I think it's a paper, very recent paper, that reports on bedding, some form of grass used for bedding about 200,000 years ago. Of course, this is still very recent, even though it's very old for organic tools. It's very recent for what we would like to be looking at. However, we do know that other types of tools used by extant primates are made of much more robust pieces of wood, and that those more robust pieces of wood, think about a branch, think about a tree trunk, those could actually fossilize in a variety of contexts. And I am of the opinion that we haven't yet found them because we haven't yet looked for them. And I can't say very much about this now because we have work in progress, very important work that may be soon revealing some very important facts about this. But I think I can say that it is expected, and it's not a crazy idea, that organic material can fossilize and remain in the record for 3-4 million years. And you can, with a lot of work, experimental, comparative, and new methods for analysis, actually make the case that probabilistically this object was likely used as a tool. Okay, so one of the big problems we have is not only about materials that we think don't fossilize. It's the whole anthropocentric view of human evolution that led to create a discipline where you only looked at human tools, even though there are a range of animals in the animal kingdom that use tools habitually, and that we only look at lithics because that's what made us special and what used to we thought correlate with brain size increase, etc. When right now the view is very different, right? You have tools that maybe 3.4 million, where hominins were still showing relatively small brain sizes, small body size, and a number of features that definitely, they were not living in the savannah, so they were living in very forested environments. So a number of features that actually is indicating that we are seeing a major shift in the overall philosophy of what is technological evolution.
Thank you. I feel bad because there are many other very interesting questions. And in fact, there is one that was talking about lithics and the bias there and so on, but you've tackled that one. And so what I'm going to do now is be slightly unfair, but I think this is a very topical question. So I'm going to ask anyway, and this is to both of you. What impact will the dramatic reduction in UK ODA research funding have on your work? And I'll start with Iruka because I really want to hear what is going on there, especially considering, obviously in the science area, there can be quite a huge amount of cost that is supported by external funding. Iruka.
Yeah, I was actually going to say 2 words about this at the end of my talk, and I thought, since I don't have a concrete answer at this point, maybe not. So the answer is, we don't know. We're terrified and holding our breath. So as you would have heard, there's some scientists in Africa who have already lost funding. This is funding that was already dispersed. So it's not reduced potential to apply for future funding, which would have been bad anyway. But the situation where people have written grants, got them, started projects, and then heard that there will be no future funding. It's unfolding as I speak. And so we're really just, listening to see what would happen. So we have my group has a diverse source of funds, but sadly, almost entirely from outside of Nigeria. The most local funding we have is from AISA, which is based at the African Academy of Sciences. And I think what Africa really needs to think about is how can we make ourselves less dependent on the politics of other countries, because this is really the result that we're seeing now. So our diarrheal disease research is funded by an MRC grant, which is currently in a no-cost extension. It will soon end. The fate of that project right now is unclear to me. We have antimicrobial resistance work that's funded by UK funding that we don't know how long it's going to continue. We're just watching. And then we have funding from other places. So we may have to scale things down. It's a bad time to be doing that because, as I mentioned now, we're getting public health institutes, we're partnering with them, we're training people, we're building the capacity that could lead to long-term surveillance. So this is actually the wrong time for this to happen. But as I mentioned, we're not choosing, because unfortunately it's UK taxpayers' money, and I think the UK electorate has to decide how that gets spent. Thank you.
Susana.
Yes, I think Iruka made a wonderful on-the-spot speech about this. I just want to say, I think it's absolutely mind-boggling that in a moment of a pandemic where science and the investment in science is being, more evident to the public, as probably it has been for 100 years, where everybody understands that without science, the survival of our species is actually seriously threatened, that we are facing, that the priorities have shifted at this stage and not shifted towards science, but away from science. I think, like Iruka said, I am also holding my breath. It is very unfortunate that we are being obliged potentially to apply for funding in other countries outside the UK to keep our research going at our field sites, potentially, where PhD students currently supported by different fellowships are being urged to hurry up to finish projects and PhDs. Extensions not being potentially met as expected, and other projects, like Iruka said, we are also working hard on capacity building together with our partners in Mozambique, in Gorogoso National Park. And I'm now scratching my head and thinking where to go to apply for substantial fundings to be able to continue this extremely important work. I also want to say a word about how some areas are likely to be hit harder than others, right? Just, the pandemic together with these cuts, it's really hitting extremely hard everyone that is dependent on field work and anyone that is dependent on data that cannot be used directly from the computer, but that when you need to go to the field, you need to collect your data if you want to make progress in your research. All these types of data are, were already incredibly damaged by the current situation, and now with this announcement, we are all very worried. And I think the younger generations that I mentor feel quite discouraged at the moment to see this trend, right? But like Iruka said, it's the public that needs to speak loud and make the right choices when it comes to, to elect our representatives that then make choices about how to spend funds. So I hope something will change.
Thank you. I mean, the repercussions of this are obviously very far-reaching and could have very long-term effects. And, a stop-and-go kind of situation where we say we're stopping now, but we'll come back next year is not particularly reassuring, in the sense that when you think, for example, about scientific capabilities, they take very long to create. And one sort of pause like that can undo all that work and display all these capabilities. And then, one might have to sort of start again and wait for a very long lag to then get back to where they were. So, I mean, I don't want to sort of flog the horse to death, but this is all very, very worrisome, I have to say. And I hope that, there will be a change of mind and heart around this topic. Anyhow, on that note, I shall bring this discussion to an end. And I would like to thank all the speakers and the audience for joining us and the very many questions. And I must apologize that we were not able to tackle all of them. But thank you very much. I have noted them all down and will be passing them down to the speakers. And if there's anybody who, for any reason, really wants to follow up, please write to us, write to AfOx with the questions to the speakers, and we'll put you in touch with them to have your questions responded to. Just to mention that next month, we also have another insaka that will be on the 6th, Thursday, the 6th of May at 5 p.m., so British summertime. And we look forward to seeing you at our next insaka. And thank you once again, and goodbye.
Thank you.
Bye.